next 20 minutes, we will be hearing from Professor Nobutaka Hirokawa about the epilepsy-related research with KIPS genes. Hirokawa is a very well-known figure in the research of epilepsy. He is affiliated with the scheme on KAU. He had lots of visits here. We value him for his great research, especially knockout stuff that he's doing with the epilepsy disorders. So Hirokawa is going to talk about KIF2 regulates postnatal active and its deficiency causes malformation of cortical development and epilepsy. It's my pleasure to present Hirokawa to you.
So, thank you for kind introduction. Today, I will talk about unique model KIF2A and malformation of the cortical development and epilepsy, the relationship with human disease. So this book is a totally unpublished one and based on the collaboration in the CAR. Okay. So let me give you a brief introduction. We are studying molecular models, and molecular models usually play an important role for intracellular transport. So this is the in vivo axons, and these days, if you use the GFP technology, you can visualize real movement or transport of the motor protein or cargo proteins, okay? So, we are asking these questions in terms of the mechanism of intracellular transport. So, we identify all members of the molecular model, named this KIPS, in human and mice. The total number of the gene was turned out to be 45. At the gene level, so that the protein level, the number could be twice more. And so most of these members transport cargos in the neuron and other cells. But I talked today, KIF2A, it's a middle motor domain motor protein, but this is very unique. This KIF2A, they polymerize microtubules, ATP dependently. So I give you a brief introduction about the function of KIF2A. KIF2A controls brain wiring. So we identify 3 members in the KIF2 families, 2A, 2B, 2C. And 2A is abundantly expressed in the brain. And 2A has middle motor domain, okay? And to study the function of KIF2A, we generated knockout mice. And this is the mutant. Mutant mice were born but died in a day without suckling milk. So what's wrong? So this is the cortex and hippocampus and cerebellum. As you see, in knockout, laminar formation of the brain is very much affected. Why this happens? So this is the, we marked the neuron, and neuron was born in the paraventricular zone and migrate upwards. So this is wild type, and this is a knockout. The level neuron at the same day. As you can see, in knockout, the migration is very slow. This caused the defect of the laminar formation. Why this happens? That is shown in this slide. This is wild type and knockout. In wild type, this is axon, dendrite, and the axon, there are short branches, but they stay short. But this is a mutant. If you like TIF2A, the branch of the axon elongate, terribly long, okay? So that caused the defect of the migration. This is wild type. In the wild type neuron, this is cell body axon. Axon extend and cell body follow and migrate. But this is the mutant. In the case of mutant, because the long axonal branch emerge, cell body cannot know where to go. Then the laminar formation or migration is affected. So why this happens? We found TIF2A has the activity to depolymerize microtubules by hydrolyzing ADP. Very interesting. So this is a summary. In the normal growth cone, and this is the mutant. In the normal growth cone, the branch at the tip, TIF2A bind to the microtubule's tip and control the to suppress unnecessary elongation of the microtubules. But if you don't have TIF2A, microtubules elongate terribly long. That causes the long axonal branch and defect of the migration. So, but we don't know the postnatal function of TIF2A. To know this, we studied TIF2A conventional knockout, and also we generated conditional knockout to delete the TIF2A postnatal. So that is because the human disease was identified, called MCD, malformation of the cortical development, which also showed that this disease also showed severe epilepsy. So it's a TIF2A is located in this locus. And these patients showed a mental retardation and malformation of the cortical development and severe seizure, epileptic seizure. So first we studied why the MCD occurs. And we analyzed TIF2A conventional knockout mice, and you can see this is a knockout and wild type, and you can see in knockout neonatal onset seizure with clown. You see here, seizure occurs. Okay, and so even after 6 hours, knockout mice failed to roll over with the coordination disturbance. Okay. So we found out the small pale body and the congenital microcephaly in conventional knockout of TIF2A and the cerebellar hypoplasia. As you can see, wild type and knockout, in knockout mice, you can see lateral ventricular dilation and a thinner cortex and the superior colliculus, and hypoplasia of the cerebellum and midbrain. So microcephaly occurs. And this is the cortex of wild type and mutant, and cortex of wild type and mutant. You can see clearly laminar defect. Also you can see in the hippocampus, in knockout, laminar defects occurs. And obviously corpus callosum in knockout is much smaller. So we studied the reason. The reason is the apoptosis occurs in a knockout mice brain here. So this is called the loss of neuron. So this is a summary of the MCD-like phenotype of KIF2A knockout mice. Neonatal onset seizure and theta cortex and superior colliculus and hyperplasia in cortex, smaller corpus callosum, and so on. And the apoptosis occurs. So next, why epilepsy occurs? The mechanism of epilepsy. To study postnatal function of KIF2A, actually KIF2A is abundantly expressed in postnatal. We generated conditional knockout. We using tamoxifen, we conditionally knocked out KIF2A postnatal. So we applied tamoxifen from 2 weeks postnatally, about for a week, and completely deleted KIF2A postnatally. Then what happens? So conditional knockout mice show severe epilepsy, as shown here. Okay. You can see severe seizure. Okay. So where is the locus? So we found both cortex and the hippocampus, the severe seizure occurs. I'll show you one video. It's a cortex-oriented epilepsy. So during this seizure, we recorded electroencephalogram by this process. Okay. And we found also in hippocampus the seizure is originated. You can see here. Obviously, these knockout mice show the severe seizure, epilepsy. Okay, so what is the mechanism? This is the wild type and the conditional knockout stained with KIF2A. And also, this is the wild type and the conditional knockout stained with neurofilament M antibody, which is a marker for axon. As you can see, comparing with the wild type in conditional knockout, there are aberrant axonal network here in this region, very abnormal. So we thought aberrant mossy fiber sprouting occurs and form recurrent network here, as shown here. That is true. If you culture granule cell, this is wild type, in conditional knockout, many long branch of the axon emerge. This could form recurrent circuit. And if you express exogenously KIF2A, then you can rescue this phenotype. So this happens clearly because of the loss of KIF2A. Then we perform electrophysiology and proved that the HeLa stimulation causes paroxysmal depolarization shift in conditional hippocampal slice culture. So obviously, recurrent circuit, abnormal circuit is formed here. That was confirmed by the staining with zinc. Zinc is enriched in mossy fiber axons. So compare with wild type, knockout, there is lots of axonal network here, abnormal network. So that another data by the YFP expressing mice, conditional knockout mice, show clearly in this layer there are aberrant abnormal network of axons. So as shown here, this is wild type, this is a conditional knockout stained with neuronal marker, neurofilament L. You can clearly see in this region, compared to the wild type, there are abnormal network of axons. That was proved by the single neuron level. This is wild type, and wild type neuron extend long axon, one axon, and several dendrite, enlarged here, cell body and the long axon. But conditional knockout, as you can see here, this is cell body, stained with the neurofilament M, axonal marker. This neuron extend several short abnormal axons. This could form recurrent circuit. Okay, this is a summary of the phenotype of KIF2A conditional knockout. So severe cortical and hippocampal focal epilepsy occurs. And that is because aberrant neuronal network formed in cortex and hippocampus. So, and also axon dendritic differentiation was affected in this knockout mice. So in summary, we've clearly identified or clarified mechanism of cortical malformation and also epilepsy in the KIF2A conditional knockout mice, and also in a patient which is affected with the KIF2A function. So I stop here. Thank you for audience.
Thank you very much, Nobutaka. Very interesting talk. One question, as we did in this session, of Nobutaka's choice. One question.
Okay.
So for the postnatal model, did you find the reason why it's only the cortex and hippocampus axons affected? What about the other axons?
Okay. Yes. The main reason is the KIF2A expression. KIF2A expressed hippocampus, mossy fiber, abundantly, and also, cortical neuron too. So in the cortex, it happens also. I didn't show that data. So you can see the seizure in both hippocampus and cortex. Okay? You understand? Okay.